Hello, Frank. Thank you for joining me today. It's always a pleasure to talk to you and to spend time with you. And I just appreciate all the effort that you've put in over the last 20 plus years helping me and Microbial Insights. And I know you need no introduction because you are globally recognized for all the great work that you've done. But for those of you that aren't familiar with Dr. Frank Loeffler, he is the Goodrich Chair of Excellence in Civil and Environmental Engineering at the University of Tennessee in Knoxville, Tennessee. The discoveries in the Loeffler lab have really contributed to the advancement of biodegradation and bioremediation, and he's contributed to over 200 peer-reviewed publications for our field. Dr. Loeffler has been an active advisor to me personally and to Microbial Insights for over 25 years. So it's really an honor for me to have the chance to record some time with you today and to have your knowledge so I can have it for future reference and to share it with others in the field. So thank you, Frank, for being here with us.
Well, thank you very much, Dora. It's my pleasure to do this, and I hope this is going to be a good session here for people to learn about bioremediation.
Wonderful. Well, let's dive in. So you've been teaching environmental courses for a long time. If you had to think of a few things that you hope new graduates will take from what they learn in your program and implement in the field, what would you say that those would be?
Oh, that's a loaded question, but let me try to answer this. So, graduates typically work on a specific project and then they become experts in this specific area of investigation. But obviously, when they then move on, maybe a position in consulting or elsewhere, right, they should understand that there's a range of treatment options that is available. And so they should be able to select the most promising approach and guide the implementation of effective, resourceful, and eco-friendly remedial technologies, not just the specific technology they worked on during their graduate time. And they should understand that there's no one size fits all approach, right? So remedial treatments must be site specific and goal oriented. And this may not necessarily be the technology that they studied at the university. And so technologies are constantly being refined and new technologies transitioned from the lab bench to the pilot scale and then to the field. And they should know about this. So they have to be on top of all these technologies that go beyond the technology that they studied in grad school. And so my hope is that graduates have the opportunity to build networks of communication with fellow students at a diversity of universities, nationally and internationally, and with other professional colleagues. And so that they can take advantage of these networks to really select the best technologies for a given problem at a contaminated site. Graduates now, they are very well trained using AI tools, but these networks that connect real people, in my opinion, will still be the basis of success. So my hope is that graduates can capitalize on AI tools, but will continue to benefit from networks between people to make real world decisions that probably are more impactful than if you just rely on tools where you can't speak with real people. I hope I made my point clear. Another thing I want to say on this topic is maybe remediation is a discipline where you alone will probably not be successful. So what you need to do as a graduate, coming from graduate school, you want to be successful in consulting, you have to be able to function in multidisciplinary teams. So people from different disciplines work together, and each person brings their unique disciplinary knowledge to the team. So it's an additive approach. Everybody brings something to the table. Okay? So people from different disciplines come together to accomplish a common goal. So this is clear that this must work for remediation, bioremediation, and other remedial efforts. So now taking this a step further, we talk about interdisciplinary efforts. So here you integrate knowledge and methods from different disciplines and you establish a new level of discourse with the goal of developing new approaches and solutions. So this is an interactive approach where everybody brings something to the table and something new evolves, something better. And I don't believe you're quite there yet. So we have understood the transition from intra-specific effort just within one discipline to go to a multidisciplinary effort, but we are not quite at the interdisciplinary stage yet. And I think that needs to happen to further move technology along. And then I also want to point out that the next level would be a transdisciplinary effort, where researchers and stakeholders create a shared conceptual model that integrates different disciplinary perspectives. So the point here is that these transdisciplinary efforts take a holistic approach to address complicated problems. And bioremediation or remediation of contaminants is a complicated problem, right? But you have to ideally apply a holistic approach that goes beyond just cleaning up the contamination. By doing so, you generate other problems, secondary problems. You have carbon emissions. And looking at all of this holistically, that's what I mean with transdisciplinary approach. And I think this is the future, and this is a high level expectation, but I have these expectations that graduates now can recognize the differences between multidisciplinary, interdisciplinary, and transdisciplinary and strive for a transdisciplinary approach that can really move remediation to the next level. But this is my hope, what graduate students can do and eventually implement in the field.
I think it's excellent advice, Frank, and I think it applies not to just graduates, but I'm thinking about myself and being more interdisciplinary and holistic in the approach that I take in general, like as a small business and trying to relate and work together and partner with other small businesses to be more holistic in how we approach the fields. I think that's excellent advice for all of us.
Yeah. And I think an important aspect of this is the recognition that every site that requires treatment or remediation is unique. And therefore, this one size fits all approach does not apply. So every site needs some level of investigation and characterization in order for remediation to be successfully implemented. And ideally, this is done in teams, right? It doesn't always have to be a transdisciplinary team, but it has to be a multidisciplinary team for sure. And then you implement a standard technology, or if the opportunity exists, right, you have a team, a transdisciplinary team, and you generate new knowledge and understanding and advance knowledge and technology while doing remediation at that site.
I think that's very wise advice. Well, one of the questions I have for you, I know that Dihalococoides is an organism that you've done a lot of work on over the last couple of decades. And we all know that Dihalococoides was a really important discovery, understanding that organism and how to quantify and understand what the characteristics of it can do in the environment, change the landscape of what we do in remediation. Is there another organism that you have found that you see that's interesting, that people should better understand or better investigate?
I mean, clearly Dihalococoides and also a partner, close partner, Dihalogenemonas are organisms that are clearly very important for remediation of chlorinated solvents. And Microbial Insights has a comprehensive toolbox, right, for monitoring these types of organisms, for site assessment, but then also for bioremediation monitoring. So clearly, these are keystone organisms for remediation of chlorinated solvents. Now the question was if there are other organisms that I find very important. Now, this is a loaded question for me to answer. Let me try. I think there are many organisms out there in nature that can have substantial impact on bioremediation. The problem is we haven't discovered them and unraveled their potential for remediation. An example, a good example may be, the problem with per and polyfluoroalkyl substances, PFASs, that currently receive a lot of attention because they emerge as global contaminants and are a big issue in many different countries. So PFAS, they appear to be true xenobiotics, meaning they are synthetic chemicals and they have no natural origins. These PFASs have been introduced into the environment starting in the 1940s. And now, from a microbe's perspective, these PFASs are potential sources of carbon and energy. My point here is that an organism that can use these chemicals as a carbon and potentially energy source have an advantage over their competitors. So there's a selective pressure for the microbes to develop strategies to use these chemicals. So there are estimates out there that talk about 1 trillion species of microbes on planet Earth. And we haven't discovered 99.999% of them. So what we know about is a small fraction, a tiny fraction. So other estimates suggest we have 5 nonillion bacteria on planet Earth. So if you don't know what a nonillion is, I also had to look this up. It's a number. It's the number 1 followed by 30 zeros, or 10 to the power of 30. My point is we have a lot. And now the question becomes, how long will it take for Earth's microbiome to evolve productive pathways catabolizing xenobiotic PFASs? Will this take 10 years, 100 years, 10,000 years? We don't know exactly, but maybe such pathways have already evolved. And we just haven't figured this out yet completely. And I will give you an example for this. And I name a microbe that brings some excitement to this field. So a colleague at Princeton University, Peter Jaffe's group, they discovered a very unusual bacterium they call Acidomicrobium strain A6. And that can couple ammonium oxidation to the reduction of ferric iron under acidic pH conditions. And under these conditions, it can apparently also degrade at least some PFASs. The issue is that this bacteria is different from bacteria that have been studied previously. So the knowledge base is very limited. Strain A6 is very challenging to grow and study. And this may be true for many other microbes that can degrade PFAS or other chemicals of concern. And now I tell you what I perceive as one of the major challenges to make progress naming new important organisms for remediation. A major challenge, in my opinion, is that students see research using computational tools as far more predictable and therefore more attractive than experimental work at the lab bench. So they become experts in bioinformatics. And this is good because the field requires computer savvy researchers that can take full advantage of omic tools in support of remediation. So this is a good thing. The downside is that we have fewer and fewer researchers who can actually discover and grow, cultivate novel microbes with high potential for innovative biotechnologies. Okay. And when I say this, it reminds me of a quote from a commentary that was published in the journal Nature some 30 years ago. It says, so many microbes, so little time. Okay. So it also means that maybe funding agencies don't give researchers sufficient time to discover new microbes, right? So we are forced to be on the fast train, the fast track, and we have to do computational work that provides new knowledge, but doesn't necessarily provide new cultures that can degrade contaminants that are then available for remediation. Not sure if this captured everything what you expected me to say regarding your question about new organisms. I didn't really name very many, but one of the issues is that we may not have discovered very many new organisms because this focus of cultivation, or the focus is going away from cultivation and rather describing the microbial world in terms of computational, with computational resources rather than with isolates and cultures.
Yeah, I think that's a really important point. And I think the current political climate, we need to make sure that they understand the value of research and development at all levels, right? From the university level and students and what they can do in the lab and academic research to what the US EPA is doing in their R&D division and identifications that are important for the future of our world. So I'm glad to hear you say that. I think it's very important and very powerful for us to remember, especially right now.
Yeah, so it's obviously also directed by funding agencies where the money is available. And right now, in my opinion, there's more money available studying microbiomes using computational tools, right? With sequencing, you have sophisticated pipelines, you have students highly educated applying these pipelines and generating very exciting new information. But at the end of the day, they don't have a culture in hand. Maybe the data they generate can give us new leads, how to cultivate new microbes and like Acidomycrobium strain A6, right? And get new information how we can actually transition this type of culture into a culture, a consortium that is useful for PFAS remediation. But often these connections from the computational knowledge back to cultivation is not pursued aggressively, and it doesn't happen. Also, people trained in computational aspects don't have the skills and the patience, right, to grow and handle difficult microbes. We lose this capability if we continue on the path we're on right now. And I don't believe this is in the interest of science and also not in the interest of the bioremediation industry, right, or biotechnology at large. We have to find a better balance between computationally, the computational approach and the lab bench approach. And that balance is, the pendulum is very much on the computational side right now. My hope is that we find a better balance.
I think that relates back to what you were talking about earlier with the interdisciplinary need that is very prevalent in all of the answers that you've expressed to these questions today.
Yeah.
Um, we've talked about the interdisciplinary approaches and some of the advancements that need to be made in our community, are there other important advancements that you think we that are, active in the environmental remediation industry, those professionals need to do, or ways we could be more effective? Do you have any advice on that aspect?
To be more effective. So the current focus is when you have a contaminated site, right? The focus is on destruction or immobilization or removal of the contaminant of concern, right? And that is based on regulation. There's a maximum contaminant level that has to be reached, and this is the primary goal. And so the current technology is focused on this specific goal. And my belief is that it has to be a more holistic approach, right? So that we can do precision bioremediation or precision remediation in general, where we specifically address the issue of contaminant concentration reduction without causing any other harm to the ecosystem. And we do this in the most efficient way possible. And that includes eco-friendly technology that doesn't release greenhouse gases, that doesn't change the pH of an aquifer, that doesn't leave anything behind, really. And I think the brute force approach we are currently using, often using, that needs to transition to precision approaches, precision bioremediation. And this transition is guided by the tools we have that have been developed in the recent decades, including sequencing, proteomics, lipidomics, and so on, all the omics approaches, together with other technologies that have advanced. And this has to be all put together in a systems approach, right? And this would transition the brute force technology, right, to a technology where we can talk about precision intervention to achieve remediation. And ultimately, this will also be more cost efficient. So it will benefit the environment, it will benefit the taxpayer or whoever has to pay for it because it will be cheaper. Obviously, the technology development was expensive, but this has been done in many different disciplines. Now the bioremediation discipline has to use all these tools that are available and apply them for their specific goals. And that can lead to precision bioremediation. And it comes back to the first question you asked, what do I expect from graduates? I expect them to be able to synthesize information and what these tools can do to move the industry along and get closer to this goal of precision intervention to achieve contaminant removal.
Yeah, I think that's a lot of it is understanding and characterizing, like you've talked about, because it's hard to be precise in our remedial practices if we just go with the brute force. It's typically because we don't have a good understanding of what
Well,
needs.
it's also what the expectation is from regulators, right? If the expectation is you have to achieve this MCL and there are no other rules really, right? This is what industry delivers. So also the regulators have to think more about the holistic picture, right? So we want contaminant removal, but we also want this and this and this. And then it's better defined. And then there's pressure on the industry to deliver precisely a solution that meets those goals. So it can be driven by the industry to meet what the public obviously wants, but maybe there also has to be a regulatory agency that better defines what the outcomes should look like. And also, one last thing I want to say here, it's very important that the public understands what the industry delivers. I'm not sure the general public really understands what consulting companies and what researchers in this field really accomplish. So this topic of outreach and explaining to people what we do is very important. I want to bring a short example here. I was told by a member of Congress several years ago that there's no money for problems that have been generated in the past. Money is available to address future problems. So legacy contamination is a past problem. So why should society spend money on past problems? And I had to think a little bit about this. And my response is very clear. We need an educated workforce who understands how to deal with contamination, past problems, because the problems of the future we are generating right now. Okay? So if we don't have a workforce trained to address these issues, we will run into major difficulties because we don't have people who know how to clean up contaminated environments. And as we speak, there will be chemicals released into the environment that we don't know about, that are very toxic and cause problems in the future. So if we don't have this trained workforce, we are generating a big problem for society. Therefore, it's critically important to continue to develop remedial technologies, give consulting companies the opportunities to develop these, to transition what academics find in the laboratory, to transition this into real-world applications. This remains critically important if we care about environmental and human health, what I assume we all do.
I would hope that assumption is accurate. And I think you bring an important point that as a society, it's important for all of us in industry, whether we're on the analytics side or we're creating compounds or whatever it may be, to be thoughtful of what we're doing for the current and for the future. So, Frank, I think you made a lot of important points through this about industry and our responsibility to work together in different disciplines and to educate regulatory and consultants and for all of us to be knowledgeable. And that makes me think about what's important as I'm trying to lead MI into the future. And so I'd like to know, what do you see? You've worked with us for over 20 years. What do you see as kind of the most important work that we're doing and the impact that we're having on the environmental community? And then if you have advice for me so that I can do better as I move forward. And I know you're on our advisory board, so I appreciate that you do this on a regular basis for me, but I think sharing it publicly kind of holds me accountable to it.
Well, first, I believe that Insights has done superbly over the last 2 decades, developing these technologies and making them available for remediation, bioremediation, and also other aspects of, other biotechnologies like corrosion and so on. I would know most about bioremediation. Having said that, of course, you can always do better. That's clear. Everybody can. And, it largely depends on our ability to develop new biotechnologies, right? And that comes back to what I said earlier. We need access to cultures that can do things better than the cultures we have available, or completely new cultures with new activities. Having just the information that these cultures exist is somewhat helpful, but not the whole story. That's one thing I want to say here. Obviously, Microbial Insights was always very fast in adopting or transitioning technologies that were developed at universities, then bringing this to practice, right? So this is not necessarily easy because practitioners don't always know what academics do. They don't always believe what academics say. And Microbial Insights was very good taking new discoveries from academia, explaining what these tools can do to practitioners and regulators, and then making them available to the broad community for biotechnologies, right? So this is not an easy task, in my opinion. And Microbial Insights was always very good at that and continues to push this boundary. So education is an aspect of this. And Microbial Insights has been very active in educating people, right? Practitioners, regulators, but also teaching academics what they need to do in order to actually transition technology into practice. So it goes both ways. And I experienced this myself, right? When Microbial Insights gives me advice, this would be of interest. So then I think about it and then, yeah, I see how this connects. So, giving advice into both directions, I think is very important. And you have done this. Coming back to education, right? The webinar series, Microbial Insights webinar series, in my assessment, is very successful, right? So I have participated and I was surprised how many people I can reach. I also talked to colleagues, academic colleagues, professional colleagues, and they tell me, did you see the seminar? Have you heard what was discussed there? So this seems to be very successful. And again, I believe this educational and outreach component is very important for this business. So yeah, I appreciate Microbial Insights' efforts to nurture collaborations between academics and practitioners, and Microbial Insights also engages in these collaborations. And I think that's very important. And it has been successful and, rewarding for me as an academic to participate.
Well, that makes me feel great. And we would love to have you on our webinar series anytime that you have a topic that you feel would be valuable.
Working on it.
Sounds great.
Yeah.
Well, as we kind of close out today, Frank, I want to open it up in case there's other things that you would like to share, or if you have a funny story or a memory, because you've worked with some amazing people in this field. If there's anything that you want to share that we could, add to this, I would love to just kind of make this open for you.
Oh, funny story. So, I mean, I'm sure there were quite a few. I'm not sure I remember all the details to make it really funny. There was one event that I remember where, you know, today bioremediation using bioaugmentation cultures is a routine technology. But in the mid 90s, early 2000s, it was not, right? So you had to explain this technology to stakeholders and the public to convince them that this is actually an effective approach and a safe approach that doesn't cause any damages or harm. And so I worked with consultants at the time, and I was responsible for the bioaugmentation culture. And the consultants referred to the bioaugmentation culture as bugs. We inject bugs into the subsurface. And I remember a situation where I explained the technology and then was asked if these crawling insects would do damage to buildings. And that was the moment when I realized that in US English language, a bug can be a bacterium or a crawling insect, what I didn't know at the time. So apparently there were indeed people who believe that the introduction of bugs into a subsurface environment involves crawling insects. And I had to then explain that no, we talk about microorganisms, bacteria. And in retrospect, I thought this was very funny for me. And after the explanation, also for the audience, when they realized that we talk about microorganisms.
Well, come a long way since the 90s, which I'm glad, but we still have a long way to go, as you mentioned today. So that was a great story, Frank. And then I
Yeah.
can almost picture a visual of some bugs crawling across the screen and that being what people are imagining.
Yeah, so I adjusted my slide deck after this experience to specifically introduce the difference between a bug with legs and a bug with flagella.
Oh, that's an awesome story. Well, thank you so much for your time today, Frank. I think a lot of the growth of microbial insights over the last 2 decades has been attributed to the collaboration that we've had with you and with your group. So I personally know that I've grown and learned a lot from you over the years and really value the relationship that we have and the advice and feedback that you give us. And I'm grateful that you were willing to do this today so we can kind of memorialize everything that we talk about for future generations to be thinking about. And so I'm grateful for your time and for your friendship and for the investment that you've made in microbial insights and actually in this industry as a whole.
Well, thank you very much. It's my pleasure and also obviously my interest, my passion. Maybe a last thing I want to add is that, biotechnology is discussed for decades as the future for technology. We can do so many things much better using biotechnology. And biotechnology certainly left an impact in many different aspects of human life. So many things have improved. But when it comes to environmental biotechnology, I think we are still in the early stages of development. There are many, many aspects where bioaugmentation with cultures that have desirable activities can have a huge impact on greenhouse gas emissions, recycling of elements, contaminant remediation, and many other aspects. We are at the very beginning. Bioaugmentation for remediation of chlorinated solvents is really the first highly successful bioaugmentation biotechnology in environmental systems. There was a recent paper in the journal Nature where researchers from Norway described a bioaugmentation approach to substantially curb emissions of the greenhouse gas nitrous oxide from agricultural soils. My point is that bioaugmentation has an enormous potential as a biotechnology in many different fields. And I believe that microbial insights can be a player, providing tools for assessment and proper implementation of all of these technologies. So I think the future's bright, okay? Because biotechnology will become more important, with the caveat that we have to have the right people who can actually culture things, grow them, and then make them available.
I agree. I think and thinking about the future being bright and what we can do and new students that will take and add to the legacy that you've left is exciting for me to think about what future generations can do with the knowledge that we've gained.
All right. And, I'm humbled that you emphasized that I was a big contributor, but all of this is a team effort, right? With lots of students and colleagues in the US and internationally. We all work together to, make this field successful. So it's not an individual. This is a group of people And I thank all my colleagues here who may see this eventually. Thank you very much for all your help and contributions.
Yeah, I feel the same. I think we work in an incredible industry with so many bright individuals, and definitely my passion. And I am very grateful for my career and what I've seen and what's been accomplished, and watching all of you that are doing amazing things to tackle some really complicated problems. So it's exciting, and I'm excited to continue and see where we go. But I'm very grateful for you, Frank, and I look forward to many more years of collaboration together.
Okay, me too.
All
All
right.
right.
Thanks,
Thank
Frank.
you.